Very good morning to all of you, and thank you, Sudipto. It's really my pleasure to welcome you all to this distinguished lecture of today. So this is just a beginning from the Department of Biotechnology and Microbiology. We'll have lined up several distinguished lectures in the coming months. It is a reflection of I think this is a reflection of চলে যাচ্ছে মাইক থাকছে না হ্যাঁ 100 Okay, so what I was talking about, that there will be several such distinguished lectures in coming months, and it is our reflection of our continuous effort to foster academic excellence in Tipthe University. Now, as Sudipta already mentioned, Professor Dwaipayan Bharadwaj joined Department of Biotechnology as a senior professor only 1 month back, after retiring from Jawaharlal Nehru University, New Delhi as a professor of biotechnology. Professor Bharadwaj is a renowned scientist in the field of biomedical science. His pioneering work in the field of human genetics, genomics, and also different types of complex diseases, including the diabetes, has earned him immense amount of national and international acclaim. If we look into today's topic, that fat genes and fat pockets, how genetics and socioeconomic status can tip the scale, I think this topic touches a very contemporary and socially relevant issues. How our genetic blueprint and different types of socioeconomic factor interacts with each other and influences the health outcome. Maybe obesity, maybe different other types of complex metabolic diseases. It's also, I believe, that example of a convergence of hardcore biomedical science research and as well as public health. So I'm pretty sure that this is going to be a very engaging, enlightening, and thought-provoking session for both our faculty members and the students and research scholars as well. Professor Bharadwaj, on behalf of the Sister Nivedita University, I extend my warm welcome to you. It is indeed our privilege and our honor to have you today for this particular session. We are all eagerly waiting for this engaging session, sir. Thank you.
Am I audible? Okay. Thank you, Amit, for nice introduction. See that obviously I am a newcomer here in this university. So I will give a talk that of my recent research. Actually, I stopped research, to tell you frankly, that I am not I hung my boot. No experimental research I will do. But what I'm going to show you today, that's just within last year. That's one of my major publication in obesity research came just a couple of months back. And when I will talk, uh, see that many of you are from undergraduates and others, so you may not understand some of the jargons, some of those telltale tales, what we are going to say, but don't hesitate. If you do not understand, but if you want to know that, ask me. According to my ability, I will explain that. And I will go slow so that you can understand more. So it's nothing like those people are fat. So in telltale situation in Indian scenario, we always think that chubby little boy is healthy. Mama is always telling that ei khaccho na kichu roga hoye jaccho. So roga is unhealthy, and when you are a chubby kid, you are healthy. That's the notion of our country. And another thing, before jumping to that place, I will tell you another difference with the Western countries. In Western countries, the economically lower strata people, they eat McDonald's. And here, upper strata people, they eat McDonald's. So if you visit or if you see in television, you will see their Native American people, those black American people, they are massive because they are economically poor in America. But if you go to the university campus, then you will see they are much healthier because they understand healthy diet and everything. But I will tell you one more thing. See that healthy food, they don't taste good. When you go to those 5 trucks there, you will get all of those tasty food. So taste this, all of those taste, 5 different tastes, actually for survival, because we can taste poison, see that thousands of years ago our forefathers, so then I will not die, actually comes back to you when we can taste good. You know? So let's start. So first, I always show the last slide. This is the acknowledgement slide. 2 slides I will show because this total of my research with human patients, and human patients you will never get if you do not have physician friends. So when I came in 2000 in IGIB, at the time I didn't know anything about genetics. I didn't know anything about type 2 diabetes. I didn't know about obesity and anything like that. I was an immunologist, cell-mediated immunologist. And all of my major paper from abroad in Journal of Experimental Medicine, Journal of Immunology, like that. When I came here, then there was a maverick director in IGIB. IGIB was not known that much, name as Center of Biotechnology, and they recruited a maverick director. So from very first day, I had a fight with that maverick director because they say I am a rebel character. But somehow, he told me, don't do animal-based research in this country. So very first day he was telling me, animal ko chuna mat, because then you will not get out of that anything, because most of our animals, that means mammalian animals in the laboratories, are fungal infected. Actually true, because later in National Institute of Immunology, they have a problem. They find out all of their animals are fungal infected. So he said, better you work with the human population. Nobody is doing, everybody is afraid, go and do that. I said, sir, I didn't have any training. And what disease? Say that I have type 2 diabetes, stress induced, work with diabetes. So I started that. I never realized at that time that it was a trap. At the time I didn't know this is trap to become famous. But it was a trap because patients will come from these people. All the patients will come from these people. This pointer working or not working, not idea. From these doctors. But controls, because you need controls, because there are studies, you need cases and controls. Control will not go to the physician. So then I realized it's a hazard that how I will get those controls. Then what I did, then I went to do the camps, diabetes awareness camps. So telling them that I will give all of those biochemical parameters you for free, you give us blood. So 5,000, 6,000 rupees biochemical evaluation I'll give the people. So in the process, that initial 5 years of my life in IGIB was a 7 days work. Because Saturday, Sunday I used to do camp, taking cars and students go to those all Delhi NCR area and doing fasting camp and collecting samples. And in the process, we have collected 20, 30,000 samples. So, how I will go back? I'm trying, but it's not going. It's revolting. That's fine. So, in the process, I have many collaborators, and Nikhil's name is always at the top, because at that time, no physician was ready to talk to me even. And our physicians are overburdened with the patients. They have no time. Only in AIMS, and when I will tell AIMS, AIMS means Delhi. Other AIMS, I don't know them. So Nikhil was the only endocrinologist, not knowing me, started giving me samples from them. So eventually we are now, he is one of my best friend. And all of those doctors, Dr. Mohan, Dr. Modhu, Dr. Sandeep Mathur, everyone provided me those diabetes samples. But the moment I started camping, then together I collected 20,000 diabetes people, and all of them together gave me 2,000 maybe. But it started with Nikhil. And I'm endowed financially with, I think I am in biomedical scientist. Amit is here. Amit knows many things, my batchmate, that probably I am the most funded person in this system because government give funding. So financial supports mainly from CSIR and some from DBT, ICMR and SARP. When I was in JNU, I got funding. And I have many international collaborators because this kind of disease, what is the frequency here and what is the frequency elsewhere, that's why there are lots of my international collaborators. And obviously members of CSR, IGIB, they were also involved in the project because CSR GIB project is a huge project. My last project was CardioMed and they were involved in that CardioMed project. And these are my lifeline. Whatever I am doing now, what fame I got, I didn't do anything. These are my foot soldiers. Without them, I am nowhere. I started the laboratory with these three: Anubha, Amitabh, Srinivas. They were, once upon a time, 5 years back, Anubha was assistant professor in Oxford University, Amitabh was assistant professor in Harvard Channing School of Life Sciences, and Srinivas was in assistant professor in Cambridge University. Unfortunately, I lost Amitabh due to lung cancer. And all of these students, they gone through my lab, but I have only 13 PhD students. Janaki is my last PhD student, now waiting for the Viva. Without them, I cannot do anything. So what is obesity? So this cartoon slide will show you that increase in fat mass sufficient that will adversely affect your health. But initially in the childhood when we are obese, see that I am also overweight, little overweight. So then we do not know what is going on. And this generation specifically, there is no ground or for play football or cricket or anything like that. From very morning I see my kids, they started computer game and everything like that, and they are getting heavily loaded food. So everything, their energy intake and energy expenditure, if you think, everything is going hayway. क्या हो रहा है? एक बार में ही चला जा रहा है. So genetic is little bit of that. These are complex diseases. There is a genetic signature. Maybe with the proper lifestyle, there will be only difference of a non-genetic signature kit and the genetic signature kit 1 kilo. But the metabolic rate is very slow in those genetic kits, these multiple gene with them, and that changes the body weight, what you are seeing. What is the basis of genetic diseases? Everywhere in the world, because in the textbook you will find, best thing is like that twin studies. There are two kinds of twins, identical twins and non-identical twins. So if you take monozygotic twins like them, and then you see epidemiologically or story-wise, they were apart. In Western countries, many a time adoption and other cases happen. So one with a rich parent and one with a poor parents. And then you find what they are going on, and then you are saying with different environment also, monozygotic twins, their BMI is more or less similar. So when you are studying with those non-identical twins and they are in the same environment and getting either high food or low nutrient rich food, you are seeing the same thing. So these are initial studies, and they show that height, weight, BMI, the correlation of monogenetic pairs are almost 1. So they are very much. So what does it mean? It means there are some genetic play is going on there. That's why it's both are going towards them. And 50 to 70% of this total variant in the BMI is due to genetic variations. But they are Caucasian population. So Caucasian population, why I am saying in India there are 4 major population. It's very important. There are 28 different population, but 4 major population. One, that Indo-European, they are staying in the northern part of India. Tibeto-Burman, looking much more like Han Chinese, they in the Himalayan belt. Dravidian population in the southern part of India. And MP area, middle part, all the tribals of India. And recently we are finding they are the original people of India. See that if I say correctly scientifically, most of us are alien in India, except those tribal belt. All of us came during that migration. So this is that years important, that whatever you are doing, high calorie intake in that from your birth to 20 years will dictate what you will be within 40 years. That dictation is the early onset of metabolic syndrome, and obesity will take you towards type 2 diabetes, cancer, or any other of those highly metabolic disorder. So bridge is important. That bridge you are creating in your earlier days, going to those 5 trucks or McDonald's. This bridge to attain homeostasis of adult life. And this is, you see that last data of this census in 2006, prevalence of overweight or obesity increased from 16% to 24% within that 4 or 5 years among urban school-going children. Why I am saying urban? Because all of my samples I collected from Delhi NCR urban area. So there are divisions, that is the socio-economic importance. I will show in my last slides that rural. We have collected some rural samples also. So this, I will not go that much. These are the study by NFHS. They showed that how obesity is increasing that total these days. Why study is important in India. Everywhere you are finding prevalence. And study importance, this is a trick. All the scientists will tell you that because if I cannot show the problem, the humongousness of the problem, why funding agency will give you fund? If I say that this only 2% in the world affected by the disease, no matter how severe that disease, nobody will give you funding. And obesity and type 2 diabetes, metabolic diseases, funders will always give fundings because 2 things. Because medicine, these happen to generally those economically stable people, so they can purchase medicines. So all the drug company will easily give you funding for conference and other thing if it is type 2 diabetes or obesity like that, because then they know where the money is going. So India, if you say any disease, rarest of rare disease, India will always be the number 2 position, because population wise China is ahead of us. I don't think such, because they have a very robust policy, almost we catch up or we are well ahead of China. So this is this entire thing, lack of physical activity, psychological issues. Whenever we are under stress, you'll see that all of us, sometimes on others, under stress. And at the time, if you follow yourself, you'll see that we eat more. So that's another reason. High calorie intake, socioeconomic status, obviously genetic signature. And then what happened? Then happened visceral obesity, because our obesity fat mass comes mainly under the muscles also, and in our tummy region. These are the places. And there is disbalance of good cholesterol, bad cholesterol. Actually, there is no good cholesterol, bad cholesterol, both are needed. Because without cholesterol, we cannot be alive, because all cells have that structure. Still, HDL, that if you have healthy lifestyle and you take fruits and vegetables lots, then 1 or 2 point HDL can be increased. So when you are stressed, then hypertension, then high triglyceride, then insulin resistance, those people will go and eventually encounter with the type 2 diabetes. And then what is going on in Indian population? I will show a little case by case monogenetic, and then I will go to that main GWAS thing. So our work, there are 3 ways. Mainly identifying genetic predisposition markers and onset of diseases in child. So that will provide the opportunity for early prediction and intervention. Because if you have a genetic marker, we cannot change the gene. CRISPR-Cas and everything, we eventually will do that, but that's a long long shot. But if you know that you have the genetic signature, from very early childhood, you will do exercise, you will take healthy food. So instead of 20 years, 30 years, that obesity kind of disease, you can easily push that back to 70 years. And very little has been explored in this regard in Indian children. Not only Indian children, children obesity is a free area for India. Because Western countries, they will not challenge you. Because in Western countries, without clinic, you cannot bleed kids. Because I need a little bit blood, it's a little bit invasive. In India, I can bleed kid with parents' permission and with the kid's permission. That is not possible in European countries. So I have less competition what I face in type 2 diabetes in childhood obesity. So that's our entire work. So it starts with those kids. So what are the catchment area? Inclusion of this entire study from Delhi NCR, school-going children of socioeconomic status, and Indo-European ethnicity, because those people stay in that area, and 9 to 18 years. And what is the exclusion criteria? Any genetic disease you do, these 2 things you will have to set first: inclusion and exclusion. Kake nebe or kake nebe na. Exclusion criteria: if kids are having chronic ailments, under medication, history of hospitalization before giving blood within 6 months, and বাচ্চা আছে না, বলবে আমার লাগবে আমি নেব না, you cannot force. So from this we collected total of around 7,000 kids. This is a little old site, and male female more or less 50-50. So this is normal weight, overweight, or obese, you know. And rural also we collected almost 700, and underprivileged kids, because what is underprivileged kids? I don't know in Calcutta that much, but in Delhi area, that privileged kids, they go to the public school, Delhi public school, Vishwabharati school, because they are, you will have to pay more. But all of those Kanna Vidyalaya or central school, everybody can go, and there is almost no fees involved. So in Delhi, I'm finding that lower economic status kids are going to those schools. So school help us from the collection of samples and knowing that they belong to those 2 stratas. And also those economically poor status people, they still play a little because they don't have computer at their home or laptop at their home. So those are these weight and everything based on coal chart, because that's the universal in case of BMI for the kids. Every 6 months is changed. That is a Bible, is that coal's chart. So everything follows by that. Without that, there is no publication. And everything we measured, because whatever we flawed in type 2 diabetes, so here I was more matured, and everything besides that, all biochemical parameters, even all of those, your interleukins and everything, TSH, T4, T3, everything we measured. You see, leptin, resistin, and then did GWAS. So when we finished the obesity GWAS, then at a shot, we have finished 26 GWAS. And some of those genome-wide association studies first in the world, that interleukin and other we did in the childhood. So those are the different stories. So before going that, we started showing association with those Indian kids. So there is a paper in Nature, MC4R. We tested that, and we find that genotype-wise, that total overweight, normal weight and obese kids, we find there is a that is also replicated in our population when we find there is a strong influence near that variation, and that dictates adiposity in our population also. We publish another one, FTO, that time Nature came with couple of papers on FTO, everybody showing that it's associated with childhood obesity, adult obesity and type 2 diabetes. We checked that also and got a paper that it is playing the same thing in our population, but in adult, FTO is non-responsible. So then simultaneously what we did simple to complex pathway. So monogeneity mein thoda hath pak gaya. Now tested whether pathway based we can do in our scenario, because these are all costly. So first thing, as I said, we measured everything, adiponectin, resistin, IL-6, NGF, leptin. What are these things, IL-6, NGF, leptin, IL-8? These are inflammatory cytokine. That means whenever you have fever, then you will have those inflammatory condition. It is well known in case of type 2 diabetes and obesity, there is a regular inflammation going on. This is a chronic inflammation. That inflammation is not like that huge, but it's 1 kg, 2 kg inflammation. And you measured by C-reactive protein. C-reactive protein is human's primary major acute phase protein. If you have a burn injury or a something, jor hua fever, then within 24 hours, that C-reactive protein jump from 0.1 milligram per ml to 1 milligram per ml. Then after 48 hours, it will come down again. What is doing? According to theory, and some of my work also, earlier in USA, we showed that this is actually helping, because B cell, your IgG will not come to foray before 3-4 days. So how your body will fight? So CRP will immediately go and cover that foreign protein with that, and then taking that foreign protein towards the destruction. So it's the major acute fat protein. These proteins are synthesized in the liver and immediately comes into your body. But if you have a low level high all the time, that is that chronic things, and it's later found all of those chronic disease has in between C-reactive protein in your body all the time a little high. So we measured all of those inflammatory cytokines and show that obese versus non-obese and overweight, it's statistically significant, because you see that 10 to the power minus 16, because all of you know a little bit statistics. So where the significant level is 10 to the power minus 3, it is highly significant all of those time. And then what we did? Sometimes working. So pro-inflammatory markers and anti-inflammatory markers, we showed anti-inflammatory markers is high sometimes and pro is sometimes. And both the overweight and obese children, they exhibit the pro-inflammatory state due to increased blood levels of these pro-inflammatory cytokines. Then what happened? So we saw there are 3 pathways. So we tested those pathways genes, IL-6, leptin receptors and PBF1, and showed they are significantly high in the obese people. And if I take, because there are 3 genes, so 3 alleles, so alleles will be 6, na? Biallelic. So if you see this chart, what we are doing, if you have 2, your risk ratio is here, 2 to 3 you carry. So if you have all 3 genes, all 6 homozygous, then your risk is increasing. And we publish that. Then we check homocysteine pathway, another pathway all the time haywire in our body. So this is a single carbon pathway. And we showed all of those genes and tested in the childhood obesity. And showed that AMD1, that adenylyl methanoline carboxylase, decarboxylase, is significantly associated in our childhood obesity kids, obese kids. And we publish. So now, people, when they are doing with the many genes, many things, they follow some hypothesis. Because what's the process of science? You should have a question or objective. And to solve that question, you need tools. Tools are those jivas, your gel, everything's like that. Now you use your tools to solve that question. So get your result. This is the process of science. This is true for every subject. Okay. So main theory was common disease, common variant. If it is a common disease, so variants will be common all over the world. But there is a effect size and allele frequency. So if the allele frequency is very common and effect size very, very less, this detection is very easy. More and more you are going towards this, you need thousands and thousands of samples. So we generally try to play here because this is very difficult until and unless globally 100 people comes together, then you have a 1 million sample. So we play low frequency variants with immediate, intermediate effect. You are not asking any questions, students, because these are some of those areas when you will do some statistics course, you will learn. So those applications you are seeing here. So there are 2 kinds of obesity. Monogenetic obesity, that means single gene, if you have a mutation, they are very rare, but very problematic. Like this mouse paper in Cell came with a cover page like that. That is the early onset. In general, 1 gene is defective, leptin or leptin receptor, and you have that kind of phenotype. But whatever our work is polygenic obesity, that is that common obesity, modest genetic contribution, hundreds of variation. So to get those is difficult. So then what we did? We did genome-wide association studies. How we did that? Now we are going towards our major work. So what is genome-wide association? You collect some cases, obese and non-obese controls. And you take their DNAs and do the same mutation studies, but those variations are on a chip. Say, you know those slides, glass slides? On glass slides, those mutations are, in my case, was 610,000. It's a hybridization base. So that area oligos, with the control oligos, company will put them there with their own chemistry and they will provide you the kit. So now I will take from sample from the obese and make the DNA and hybridize with their technique there, and then I will see which is lighting up, because it's biallelic, whether it's A or it's a T. So then I will know exactly what is going on. It's very costly, but this is that chips that we do not make commercially available. Earlier only 1 company was making chip, now 2 companies are making the same chip. But now after new generation sequencing is coming so cheap, so people are not doing GWAS because they will do thousands of human genome sequencing, so you will get much more information. So this is the thing, 2007 this technique comes, and by 2020 you will see lots of people are doing that. But thing is like that, all of these populations are mainly European Caucasian. It's not American people also, it's not other places also. And this is that very least representation other than this European. So there are non-European only 5 GWAS being done, Asian population not studies. So our study is the first one for this population in India. Childhood obesity, since I already told you, this is Indo-European ethnicity. So what we did, we attacked in the two way, because these GWAS, these variations are intragenic or intergenic. Mainly 99% of this single nucleotide polymorphism, this ATCG, are falling in the non-genic area. But you understand linkage, if I get that, so I know it linked with the what gene, and with those techniques I will find out the gene. But when I am doing these rare variants, these are the functional variants. These are the codon changing variants, or the 5' upstream, that Tata box, your transcription factor binding sites, those variations. So they are the functional variants. We did the same thing on GWAS and XWAS. What is the fun with that? If I do GWAS, I know the address. And if I do the UAS, then I know the cause. Instead of going and doing that myself using recombinant DNA technology. Now I overlap this one over this chromosome. So in one shot, I know what are the functional differences coming on because of that SNP. So we did in this case what we didn't do in case of type 2 diabetes, because we didn't know this exists and we can do it here. So then what we did, we finished with the samples. We used the Affymetrix axiom array. SNPs are, this is 675,000 SNPs, and fast phase 1,500 urban private school. And then we got all of those association. We tested them in the replication phase in 4,000 samples with those main tested 1,400 SNPs. And here we added rural sample, government sample, urban government sample, total those things to see what is going on, because my title says that there is a socio-economic bias. And we did the meta-analysis, and here we took the 5,000 and did the functional variant analysis. This is the statistical jargon, and anybody is interested, you can contact me. I will teach you how to do that. So this is that in one way, the entire GWAS. What is the way? Sample collections. I told you this is before, all Delhi based. And then what we did genotyping, and then we did sample QC. This is quality control. When you have any data, you will have to do quality control. You have two things, samples and those chips. So you have those chips, SNP QC and sample QC. So based on missingness, heterogeneity, because when you are doing SNP QC, you can get wonderful ideas. You say that your sample says actually it's a female, but you are seeing Y chromosome coming here. So you throw that away because somewhere you did a mistake. So these are the QC is actually your mistake checking also. And then you use Hardy-Weinberg equilibrium, cryptic relationness and everything. So then you do discovery phase. It looks like why it is called Manhattan plot. You know, New York, Manhattan, from distance it looks like this. Not to be afraid, every color represents one chromosome. Chromosome 1, chromosome 2, chromosome 3, chromosome 4, all the 23 pairs of chromosomes are there. And in this case, these are SNPs. This is the cutoff value. Over there, you will find those SNPs. Those SNPs are your important because after analysis, they are showing up. You take them. There will be some negative control, negative and positive signals, but that will be measured in the replication phase. And then you integrated those genes in the regulatory information. This is added because we did both these GWAS and UAS and association of the signals to metabolic phenotype, and then to the pathway analysis. And after that, what we did transcriptomic analysis to show that those variations exist or expressed in the cells or not. So when we did obesity, you see, we started getting new novel thing. Itna novel मेरे को type 2 diabetes में नहीं मिला because he has a less competition. And then we did that BMI, GWAS, we got 3 new variations. Weight, we got 5 new variations, 5 new genes. But obesity genes, none of those genes we got, novel genes, that cross that statistical barrier. Then waist circumference, we got 5 new genes. These genes never in the world being tagged with the obesity pathway or obesity relation. This is our giving to the world. And then hip circumference, 1. Waist hip circumference, we didn't get 1. Now you will see, because when we do experiment, we have some controls, positive controls. If positive control doesn't work, that means something wrong in your experiment. What's in genetics positive control? See, this is I'm teaching you students. You see this SNTG1? So this variation is significant in case of hip circumferences. But it should show here also, not with that significance, but significant level. You see SNTG1 here also, SNTG1 here also. So if it is a false positive, it cannot be everywhere. And then you show the pathway if it is known that what pathway and how metabolically it is related. So this is another way of showing that Manhattan plot. It didn't come to my mind. That's what I'm saying. The students are cleverer than us. Students are much more intelligent than us. This is the Janoki, that my last student, current student, because she was writing the paper. So I told her that we need to give different representation. You know why? That probably students will not know now, because there are journals. There are nature genetics, science, and there are chota mota journal, there are mid-level society journals. So what make them this? If you go and check nature is a journal like those anybody can read. Any of one of your student, if you start nature, then you will find something, some news or something very interesting science-wise. It's not always the scientific paper. So make it a habit to read that, you will learn many things. You will see nature papers never come with a straightforward figure. It's a different figure. The same representation, because their pages are very costly. That means for a full paper, they will give 2.5 pages. You will have to accumulate everything with like that. That is that slide making procedure. One class I took with you, I told you that every information thoda hatke. So she saw there, and then it's entirely her idea. She didn't pick up for nature. She make that thing, mera dimag mein nahi aata. That you see that all of these are 1, 1, 1, that chromosome, 20 chromosome, and these are the traits. See, in one picture, everything together. This is obesity. So all colors are there. This is BMI, this is weight, this is waist. And these are circular way, 1, 2, 3, 4, all the 22 chromosome, because we do not touch that X chromosomes and Y chromosome. Analysis we will do later, because these are all autosomes. And every chromosome, he showed that which is that marker is coming high. So I love this thing. And she showed in population, these are SNPs. What is the percentage? See that normal, neutral SNPs, we have many. All of our cytoskeletal structure SNPs, genes, and everyone has the same. So their common variants are 50-50. Whenever it's going towards the 60-40 and other, that means some selection pressure is there. Something is going on in that population. That like those, if you go to the malaria-infested places, you will see some of the genes, some of the variations are much higher. That means that is for natural selection, is giving protection from malaria. So we showed that how biased these genes in our population also. That fits with our this thing. So end of the day, we got 6 novel variants, novel genes, and 2 well-reported variants. So this too is important, because it gives you all the time strength and stamina and belief, because you are also replicating something other. If I get only novel, that means I also have some problem, because it cannot happen that in America some genes are there, is not replicated here. So then this is the meta-analysis, that all of those numbers, these are the numbers of samples. These are those genes we showed in those genomoid, that circular picture. We are now showing discover phase mein, what is that significant level? Same gene, what is in replication level, this significant level, and if we add them, that is called meta-analysis, it increasing. So we got these 8 variation with those different level of 10 to the power minus 8, 10 to the power minus 11, and they are all important. They are controlling this phenotype in our population. And then what we did? One by one, we did functional studies. TCF7L2 is a transcription factor. So there are paper already, they showed that what they are doing is involving in some sort of obesity, but our SNP is not linked with them. Ours is new in a different genic region of TCF7L2. It's a huge gene. So in conclusion, TCF7L2 is not a risk factor of obesity in European population. It's never showed European population the risk. But its effect on type 2 diabetes is modulated by obesity. They showed in type 2 diabetes. In our population, this is the TCF7 gene, and our present study, this is the SNP. This is the SNP. These two are novel SNP and linked with the different genic region, and that is showing all of those 3 in our population is just changing the status of obesity in our population. So what is DAS? We took that thing and we showed that there is a variation in European population frequency and our frequency. And we checked and we find that this particular genic region is interacting with 2 different JDHH6 and GPAM. What they are doing, these are the genic name, how they are modulating. We took some bioinformatics and we find out before going to that experiment that JDHH6 is actually a zinc finger. What is a zinc finger? Zinc finger motif, mainly they are transcription factors, okay? There is a motif. So it is actually controlling palmitoyl transferase. So what is that? GPAM, it's help in glycerolipid synthesis. Triglyceride is a parameters of the obesity, triglyceride synthesis. So what we get actually correct thing. That this is the right gene we got, because biochemist, wo kisi ko choda nahi. Go to the Lellinger, you will see that all of those genes care function is written there. Now the gene hunters like us, we are looking for those genes and going back to the Lellinger finding, ये तो बोल के गया, यही तो है उधर में, ये तो पूरा जो हम उसका gene उसका पता नहीं था, but we find out the gene. So IPMK, you know, ionocetyl phosphate multikinase, it's a signaling molecule. All of you are interested in molecular signaling, is a major signaling molecule. So we got there are 2 SNPs and both are related with BMI and hip circumference. And population wise, 35% in our population versus 65% of our population. What it does, is already shown this IPMK, this pathway, if you tinker with that, then you will have type 2 diabetes or adult obesity. So there is a transcription factor binding site. In any textbook, you know, this is that, these are all transcription factor binding sites. So this is altering this site. So there is a hypothalamic POMC neuron which is actually affected, and leptin melanocortin pathway of obesity is tinkered. And that is that thing, long ago, this was the cover picture of cell. That OB mouse and OB negative, OB negative mouse, we got that in human. So this is also that food intake, hypothalamic, because whenever I say obesity, people are thinking obesity or type 2 diabetes, according to me, is a brain disease. See, if a lion kills, lion eats, bagh bhi kha lete hai. If you see after that, that tiger udhar mein baitha hai, jungle mein ghumte ho, to you can see that usko samne se bhi wo horin, deer is moving, he or she tiger will not bother because it's full. But we are after lunch, we go to a meeting, and then meeting is helping us, faculties with lots of cashew and everything in front of us. We start nibble with that. But it's not like that we are hungry, but after lunch the meeting starts. So that is extra energy like that. So that kind of thing is happening. Hypothalamus is telling, oh, acha khana hai, khale, free mein mil raha hai. So we are doing that. So energy expenditure in a meeting room is 0. So we are doing, and actually leptin and disbalance, leptin को disbalance कर रहा है, that उसको निकलना जरूरी नहीं था, अभी वो निकल रहा है. So another one is the inner mitochondrial membrane peptide subunit. What it does, that is also see that huge discrepancy in the population. One variation is 33%, one variation is 67%. होना चाहिए था 50-50. So something is going on. So that's the expression QTL. That we find that not only a mutation, that's the expression QTL, because that base changing is changing the pattern of expression. What it does, it's a homodeficiency of mitoribosome protein, CRIF1, and that goes to making those PMSC. So when it is going, there is an unfolded protein response. You know that protein, when they are making in the body, so how they are taking hemoglobin shape. वो तो जीन से RNA, DNA, your protein सब हो गया, replication, transcription, then that protein should be a straight chain. So who will make this thing? There we know those folded protein response and unfolded, it happened inside the cell before coming on, and those areas are very stress areas. There are all of those, if you give the stress, there are lots of paper, they showed that wrong protein folding. And what is that common thing you know that protein folding wrong? Students, can you tell me? Are dur baba, yeah, every day in house you are doing that. Alzheimer. हाँ, faculty को बोला. अंडा को देखा ना ऑमलेट बनाने में. So वही तो है ना, that different shape ले लिया ना, वो तो protein है ना, that's the easiest this thing. So during this IMPL response, if that mutation is there, this unfolded protein response is going haywire, and you have this thing. These are the paper already published. So that is that unfolded protein response in PMNC neuron. So when I have a mutation here, is causing that stress. So there a stress protein will be there. And I told some of them I measured. Here I will tell my loopholes. Because all of these genes, we did transcriptome in the cells. But what cell? Because this time we are a little mature, so we collected little RNA during the collection of the samples. But maybe blood is not the right tissue for expression. Genes are in everywhere, but expression is not everywhere. Na? Your pancreas only expresses your insulin, but insulin gene here also, this is not expressing methylated. So at that time we collected some samples and we checked those expression profiles. Here it is obese versus non-obese with 6 samples showing wonderfully that what we did the right. And then we showed that meta-analysis, it's strongly correlated with the weight. And what it does that if you have in musculoskeletal CDC5L, then you have this TT-TA or AA variation, and then you will find that there is a chromatin interaction pattern is changing. It changes, there are 3 different pathways, that GPR116, that is insulin-mediated glucose uptake in adipocytes, RANX2, that is down-regulated the adipocyte differentiation, and DLK2, it regulates the adipogenesis. And all are actually related with CDC5L. So it's controlling the 3 different pathways. So tinkering of that mutation is causing trouble in all of those 3. Expression profile, we checked in 6 samples. You see, normal and obese, there are expression pattern. You may say that, sir, that is not the right tissue. That I said that it is not, maybe, may not be, but blood is the reflection of everything. You are sick or you are good, blood has all the time something. So we took chances. I cannot go and test liver katke or other, I cannot do that. So that is that regulatory effect on the adipogenesis or adipose metabolism, that controlling. What is that SNTG1, syntropin gamma 1, the pehle dekha, that changes the glycyl glycerol to phosphatidic acids, that ATP to ATP, and this is the syntropin work in that area. And this gamma syntropin that regulates already known the subcellular localization of DGK to ensure the termination of glycerol that signaling. That paper is already there. This is the paper, JBC paper in 2001. So what is happening? MX1, that one of those 8, it shows that is also expression and it's different in thyroid and musculoskeletal, and it showed whenever it happens, it changes the splicing site. That means splicing job hota hai, kya hota hai? Then different protein nikalta hai. So if you have a mutation there which is changing the splicing pattern, so in normal circumstances body required A protein, so now with this protein it changes to the B protein. So that happened, so entire chromatin interaction with that particular gene goes haywire. That 2 pathway, regulator of the glucose and lipid metabolism and association with the BMI and triglyceride, both are changing. And that is the expression pattern. You see, there is also change in the expression pattern of those genes. So what I showed all of those 6 genes. Now I am showing which tissue, because I told I only checked with the blood. But you will tell that, sir, why it is blood? So commercially available tissue panels are available these days. They are very costly and they are single sample. So we purchased one and we showed that you see all of those 6, all of those except one have highest expression in the adipocytes. Only 1 or 2, they are not doing the pattern because they are also in the adipocytes and most of them also expresses in the brain. So this is the tissue expression of that, all of those tissue available commercially. We tested that and we checked, because these days the internet, there is a site, I forget the name. They are every tissue, whatever from anywhere in the world they are finding, they are putting their data there. So you know, instead of one tissue, there are 1,000 samples are there. We checked it there. So what is going on? Now, I'm on the last phase of my talk, 2 or 3 slides. So you know that another hormone is giving you trouble. That is the thyroid hormone. And it's already, we know, thyroid hormonal status dictates the obesity. So when we did that, our sample has that. We checked. There are T3, T4, TSH3 measurables. But we find there are correlation with the T3 and T4. TSH is not showing that much correlation when I'm taking the samples of normal weight, overweight, and obese. But these two, T3 and T4, we are saying that reasonable sample, 2,500 and 5,000 samples, we are showing that. So collectively what I showed, that if I say obesity, how it happens. This happens from hypothalamus, PMC neuron, leptin metal melatonin, this thing, and becoming obesity. Or if you have a thyroid hormone, then you can have obesity. Or if you have unfolded protein response, you have obesity. Or you have a problem in adipogenesis, lipid synthesis, triglyceride, you can have obesity. So this is that linear pathway. And you place our genes. You see, for this POMC neuron, we got 2 genes, unfolded, 1 gene. Adipocyte regulation pathway, we saw that CDC5L, splice QTL MXC1, promote TCF7L2, the transcription factor, and SNTG1. And AKT pathway, we got one kinase, MX1. And thyroid hormone also controlled by the same kinase. So childhood obesity genomics, what we have shown, that my slide almost done. So the moment we finish this BMI or weight related GWAS and functional studies, that moment we finished 26 other GWASs. Those papers also we published, but we are not going to tell them here because those are in one shot, only analysis. So here that title, worldwide trends showing that underweight and obesity within these 3 decades is huge. There's a Lancet paper couple of years back. It showed that double burden in India, we are showing in some South Asia and parts of Africa, they concluded
that it's not like Caucasian problem. Its problem in Africa and South Asia is double burden. That your nutrition is come, you get a different phenotype, and type 2 diabetes, which is lean. And you have high nutrition, then you have obesity and type 2 diabetes of the obese. So it's a double burden killing South Asia, all the poor countries. So India also is a poor country, South Asian country and your African country. So what is going on? We check that. Because I told initially we collected from Delhi, private schools and government schools. And their socioeconomic status, different. We checked the urban government school and rural school, private. And we measured their physical activity calorie intake. And we showed is that what way they are going. And then when we check the age sex adjusted BMI, we see that urban private is always high with the BMI status. That means motapa jada hai because they have purchasing power of McDonald's. They do not. And prevalence of underweight higher in rural school, that double burden. And prevalence of obesity higher in urban private school. Exactly Lancet what they said is clearly we are finding it here. So we checked some of those, how the BMI vary with the socioeconomic status. You see that F2, all of them are follow the same pattern, but urban private is the highest. So gene is there in rural, in urban, government, that lower socioeconomic status, and obviously urban private. So you find the trend is almost same, except one or two, maybe that is that I will not conclude anything, because number 10,000, 20,000 will change that, but that changing patterns of the BMI, if they carry that genetic signature, is the same. So individuals with the same genotype, urban private school children had the highest age and sex-adjusted BMI, but the pattern is same with the rural kids also. And how does BMI vary with those incorporating genotype data? If I put the genotype data, that is that measurement, you see that same pattern it following. So what you are seeing in the phenotype and what you are seeing in the genotype, they are matching. So in summary, what I showed you, this is the last slide, candidate gene studies, they identified F2 and MC4 are already known, we replicated. Pathway-based studies, already known variations we checked. Inflammatory pathway genes, IL-6, leptin, PBF, homocysteine pathway. Then we did GWAS and we got 6 novel variants in 6 different traits. And what we did, we showed that one variation controlled triglyceride levels, one mutation controls unfolded protein response, one controlling the AKT pathway, signaling pathway, one adipocyte biology controls, and one controls lipid synthesis, and one is a splice QTL variation. And also we showed BMI varies with the socioeconomic status, and children from urban private school, they are always at high risk of being obese compared to other school types for the same genotype. So thank you. And because this data, this Indian genetic data, not only India, because besides Europe there is no data. Okay, not even type 2 diabetes, not even here to total blank. So there are global people, they want to see, eta to hobe na, that this genes is working in this population, this genes is different population, pathway of adiposity is same everywhere in the world. So they want to see, find the minor variation which needs 1 million sample. So there are people make a consortium. They are mainly white groups, so they are more powerful. So then they ask that initial we got these genes, so give us the summary statistics data. So we analyze and give them. And those papers are coming in big journals, that every year my students are getting at least 2-3 Nature Genetics, this kind of paper because of this consortium, because entire South Asian, only we have the data. So that's the added benefit my students are getting, because before going abroad, they already have their name, every one of them, 2-3 Nature Genetics and other. And after going there, they definitely get bigger labs, and from their independent lab, they always publish either Nature or Molecular Cell or Nature Genetics. So if you have any question, I'll be happy to answer. শুনতে পাচ্ছি
frequency in other population as well? Like in Indian population, we found 6 Nobel. So is there also common in European and African regarding the allele frequency?
See that nobody asked me for those variations, and see that whenever I publish this data, there is a norm that all of my data I put in that significant genes there. So many of those genes they got with the minor significant level, or in the pathway they got, they didn't get, and some of them are totally novel. So that actually I answered you before that in any other way. See that always this is the positive control, like that all the genes are related to some way in the obesity. So that's a one check. Or that one I told you that in my case I got 10 to the power minus 11. Maybe the significant level in Caucasian people is 10 to the power minus 3. But we are starch eating people, they are wheat eating people. These are gene environmental interaction, they are not monogenetic disease.
Okay. So I have another question. In the
Please.
GWAS run, in Indian population, I'm curious to know.
Huh?
So in Indian population, in your GWAS, so what are the covariates you used? What are the covariates you used?
So what do you mean by covariate?
Covariates means the variates which generally affect the outcome. Generally, we use the age square, sex. So, did you adjust our principal?
So these are first slides I showed that they are all age adjusted, sex adjusted, everything adjusted, and that's why we did only with the autosomes. We didn't show the data of sex chromosomes.
Okay. Thank you.
These are any statistics in any disease will do. These are the sample QC. It will automatic.
Okay. Thank you.
I think I will have not to face any question. Please.
Sir, thank you for this wonderful lecture. Sir, as a student of nutrition, I want to know if you can suggest what kind of intervention studies can be carried out.
Intervention, how I will tell you, that's a doctor's question. The doctor will find out that what they are lacking. See, I know that earlier it was fortified food with iodine. So that kind of thing that I have no idea. Like those, I did vitamin B12 also, and I see earlier I published a paper that vitamin B12, any nutrition is rampantly low in Indian population, first thing, anything. Like I say that vitamin B12, I see that it is much lower than the Western population, and even with the male and female, male has lower. But when I'm working with that and when I discuss with those other people, that we measure the Western measurement, Western standard, which may not be true here. Because thousands of years we are here living with that amount of vitamin B12, that amount of vitamin D without those diseases. Now, see that I'm giving you one example that folic acid is important important in the case if you have low folic acid, then pregnant women will have the spina bifida and other those things. But here in India, with all of those nutritionists and all of those physicians, because this neural tube within 2 weeks or 3 weeks is completely folded, and all nutritionists and all doctors are giving high dose of folate after 5 months of pregnancy, which actually help them to go towards the future cardiac events. So in one thing I'm telling that those Western part is not fit here, nutrition way, and other things is like that. We will have to do our own measurement in 20,000 samples to show what is our own standard. And actually I did a surrogate paper 4 or 5 years back, because 20,000 samples I measured all of those biochemical parameters, and those only biochemical parameters, whether our biochemical parameters adult is correct. So when we did that, except micronutrients, measure में कोई फर्क नहीं है जो Western counterpart and others, but minor में थोड़ा थोड़ा फर्क है. As I said, vitamin B12 हमारा डाउन है, मेल में आउट डाउन है, फीमेल में उसमें कंपैरिजन में ज़्यादा है. And what is the possible cause? Because here still female childs are neglected. So अच्छा खाना मिलते ही वो लड़का को खिला देता है. So now this girl neglected, वो बाहर में जाके अमरूद ये and vitamin B12 मेजर आता है बैक्टीरिया से. So हाथ में उसीलिए वो जो बार बार सफा करते हैं ना, कोविड हैबिट इज वेरी बैड हैबिट. When COVID is that fine, because your normal vitamin B12 coming with your dirty hand. वो नहीं है एकदम ये भाभी जाबी लैब वर्क करके खाओ, वो तो not that, but little all the time hand washing is not like that. एक समोसा मिला खा लो, because बैक्टीरिया से आता है that vitamin B12.
Thank you, sir. First of all, it was a very interesting talk. I have just one question that whenever you were studying the variance in the tissue level, for one of the variants, that is IMMPL2, the result was that other than the adipose tissue, in the liver tissue, it was the level was very high. Now, as per our knowledge, that it causes the unfolding of the proteins mainly. Now, so is there any correlation that this high level of IPL2 in liver?
I do not know that, but liver is that most of the proteins are synthesized in the liver, so it's obvious that kind of folding or unfolding thing will be there. So their presence is also required there, na, because folding to udhar mein ho kya aega. So it will go to the ribosome and have those folding and come back.
And as well as it is high in adipose tissues also. That is not that only the unfolding problem is taking place in liver. It is in the adipose tissues also.
But exactly what is doing functionally, I cannot say that. I think then it's done.
Sir, it was a fantastic talk, and I think it's very hard to accommodate all those years of work in 30-40 minutes talk. Anyway, so I do not have any deep science question. One thing, so it seems that you have correlated one thing, that those kids which are going to the government school, they are poor and they are taking all those foods which do not have much nutritional value, and the opposite thing. Don't you think it's a very oversimplified assumption?
Yeah, see that I didn't tell that other story also, because it's oversimplified. That's what I said, that poor terminology, how we will do that. So then I checked, because I didn't tell you the story of the kit sample collection. I told you my friendship with Nikhil. So they're doctors. So there was something going on with Brigadier Dr. Marwa, Nikhil, and another person. They were doing Delhi government's one study. Delhi government study was like that, going to school, all of those schools, and checking the health parameters of kids, non-invasive, nothing. So they were going and checking those things and putting those way. So first thing is that, and then Nikhil, as a friend, told me, "अरे ये हो रहा है, तू आजा, तू सैंपल वो जब लेगा, वो ethical permission लेके आजा, एम्स का मेरा है, वो देके तू blood ले ले." So that's the start of this point. But they were measuring those things. What they did, they use that government of India, these are state things. So government of Delhi is a demarcated rule that in the Delhi map, which is the agriculture rural region, this is the this region, this is the this region. And they did the socioeconomic, these things, how it comes. They say that they took some of the parameter. घर में fridge है, घर में TV है, घर में computer है. So they make that parameters in that way, then tick the boxes, and then they put that in those three economic background status. And I'm using that thing. So in my case, yeah, because what they did that, because what they did and they published in the government document, that is. But I agree with you, when I went for the rural collection, Bulandshahr and other, when I went those schools, and when I go to the Sarvodaya Vidyapit in Delhi, it's different. But still, that my attendance बच्चा JNU का अंदर में जो केंद्रीय विद्यालय है उधर में पढ़ते हैं, ठीक है? So my salary, my attendance salary, both are coming from government's coffer, are not same. So those are the parameters only, because if you say that deep things, I do not know. But those are the parameters to put them is the socioeconomic. But ये लेके चर्चा हुआ that whether I will say socioeconomically weak or they are poor this thing. So they said that वो जो Bloom अभी लिखेंगे ना, two or not two like that. So वो Bloom वो भी खड़ा किया था that don't say they are poor, don't say they are put under carpet, लिख them socioeconomically weak. So वही कर रहा हूँ पेपर करने का लगे. But if you say if I take 100 sample, उसमें 5 ऐसा भी है जो मेरे को इधर से बेच के उधर कर देगा, because वो जो भी जाएगा मेरा पता नहीं है. But we statistically, if I take 100, maybe 80% of them are putting my socio-economic background, but I do not have any capacity to go and find it out. And then the parameters I am taking school, because you or me, we will not send to the Kendriya Vidyalaya or Sarvodaya Vidyalaya my kids. जितना भी पैसा चाहे वो मैं DPS में भेजूंगा.
Okay, thank you. And last one is, I know that you are a basic scientist and you do not care much about the application thing, as I presume. So, so you have identified 6 variants or 6 genes, etc. So, what is your idea? How those could be used for any therapeutic purpose?
ये अच्छा क्वेश्चन बोला, मैं स्लाइड देखा है नहीं. See, this is complex disorder. You people understand this question, or तुम silent spectators हो? I don't care of these things. I am happy. मेरा पैसा नहीं चाहिए. मेरा जो salary मिलता है ज़्यादा. I am trained to publish, see, observe, publish papers. बाहर में जब था 2 बार पैसा मिला, क्योंकि मैं बॉस का 2 काम कर दिया. अच्छा सा मिला, एक बार $30,000 और एक बार $40,000 एक कंपनी का काम कर दिया. बट स्टिल पैसा मिला, वो खा पी के उड़ा भी दिया. बट नेवर थॉट दैट आई विल गो इनटू दैट लाइन. बट if you do a monogenetic testing like those Down syndrome and other, immediately you go to those diagnostics. Here problem is like that. Suppose that I'm coming with the diabetes type 2. Diabetes type 2, दुनिया में मेरा paper, because I also got a novel variant, TMM 163. So दुनिया में more than 144 genes responsible for type 2 diabetes mutation दिखाया. So if a person, इसका एक पेपर मैंने मेरा स्टूडेंट जानोकी को करने बोला, because जितना he who dies with the largest number of paper who wins. हमारा देश में जलाने का टाइम में वो पेपर देके मेरे को जला देगा, मेरा एक खर्चा बीबी का बच जाएगा. So what she did, that if you have those 144 multiplied by 2, 288 alleles, all you have, what is your risk? That calls odds ratio. Complex disorder in its 10-point scale. If I have factor 9 or factor 8 mutation, क्या होता है? Who can tell me, students? If I have a factor 8 mutation or factor 9 mutation, मेरे को क्या disease होगा? मेरे को चुल्लू भर पानी चाहिए. That we have those hemophilia A or hemophilia B, King's disease. That is the first known genetic diseases. Comes from the England, one of the family members. That is the factor 8, which is a huge protein, but that is a factor 8 is the only one protein in blood clotting coagulation pathway which is not protease. Baki sabhi protease and they are calcium dependent protease. Factor 9 is a calcium dependent protease, serine protease. So if you have a mutation there, if you have a mutation, you have hemophilia. 100%. That means 10 out of 10. That's the odds ratio. You have the mutation, boys, females are the carrier because they are X-linked. Okay? So if a male has that mutation, we'll have hemophilia. That means odds ratio 9.9 or other. I'm not telling 10 because there are 1 or 2 abrupt cases. They have a modifier gene they showed, it's not showing that way. So almost 10, you have the mutation, you have this thing. But if you have 288 alleles together, still you cannot cross more than 3. So that other 7 unknown factors are there, epigenetic factors, chromatin remodeling and others, because I did extensive methylation studies also, I didn't show that methylation GWAS probably globally I showed first. We showed that methylation is much more potent. If I check methylation, type 2 diabetes detection will be better. But still, वो देखे भी 10 में 5 तक पहुँचोगे. So where is the 5? So diagnostic, if my diagnostic is not 99.9% accurate, nobody will purchase that diagnostic. Who wants to know that 2 things, 2 diagnostics kabhi nahi bikega. Ek this, that if you say that 50%, you answer 50%, who wants to know? Another, that diagnostic will say that you have a lung cancer gene. Nobody will do na. मेरा है तो मैं मर जाऊंगा ना. तो who wants to know that? वो तो डर में और 10 बरस पहले मेरा एक्सप्रेस हो जाएगा. So that diagnostics, even with this whole genome sequencing also not coming until and unless it goes to we identify the entire thing, because वो कभी नहीं होगा, because stress is the parameters. जे आज बॉस क्या बोला, मेरे को उधर में पुलिस पकड़ेगा, ये जो सब स्ट्रेस है, because one of the main reason genes तो बहुत दिनों से है. So अचानक this flood gate open हो गया diabetes के लिए, why? Type 2 diabetes के लिए. 2 things. One thing is like that before 50 years, हमारा even Akbar 50 साल में मर गया. So richest person in the world. So हमारा आयु बढ़ गया. So the moment we are jumping from 37 years lifespan to now 80 years lifespan, all of those diseases were there. Those late onset diseases we are coming and seeing. This is that one thing. So until and unless we have that, nobody will purchase my diagnostics. कौन लेगा मेरा diagnostics that मेरा 10% chance है, 10% भी नहीं है type 2 diabetes होने का.
Thank you, sir.
Thank you, sir, for the fantastic lecture. So now I would like to request our Assistant Director, Dr. Rajat Pal, to give the concluding remarks.
Hello. Yeah. Yeah. So, good afternoon, everyone. Yeah. It's a wonderful talk. And what I was thinking over there sitting just before the lecture starts, when I joined my PhD lab, my PhD supervisor showed me a rack. These are the PhD thesis, what I have guided, and you took one by one and start reading. So that was the day when I started thinking or reading about type 2 diabetes. Because my PhD topic was somehow related to type 2 diabetes. And wonderfully, so many things are related to type 2 diabetes. And There is a never-ending things to discuss about this type 2 diabetes. So thank you, Professor Bharadwaj, for giving so enlightening talk. And for the students, I'm telling that this is a, nowadays, actually the regular classroom teaching is not enough, okay, for having a holistic approach to the education and academics. So these kind of interactive lectures and seminars we usually organize in our department at SNU. So for students, it will be very beneficiary for attending and listening to these lectures. So as a concluding remark, just I shall say that in the next this kind of meeting, we shall again meet with a remarkably good lecture from any other scientist. Probably we are going to have Professor Mukherjee's talk in the next meeting. So for the students, I am suggesting that attend these seminars and lectures so that you can increase your knowledge outside the classroom also. And for all the faculty members, I am thanking that they have got the time from their busy schedule to come over here. And that's enough for today. And again, we are going to the classrooms for having the regular classes and lectures from the teachers. But meanwhile, sometimes we shall again meet like this in the seminar hall to discuss something apart from the regular curriculum, but still related to the science. Okay, so thanks everybody. And let's have an end for this session. And we shall wait for the next session. Bye and goodbye.